So the next speaker is Aaron Martinez. Aaron is a geobiologist and a geochemist. He received his PhD at UC Riverside and is currently a postdoc in the Institute of Earth, Ocean, and Atmospheric Sciences here at Rutgers University. And he's going to give a short presentation on biomarkers in the geologic record. Aaron, it's all yours.
Thanks. This is going to be a little bit different than some of the previous talks and most of the following talks. Think about it like more of an ENIGMA support talk rather than like a full protein talk. And so what I'm going to talk to you about is like kind of in a tangential vein to what we just heard, which was fantastic, which is not life is what, but life was where. So where exactly, or how do you find evidence of life on the planet, more or less? And throughout the history of the Earth, there have been a lot of different chemistries, global climates, and sort of catastrophes that have befallen the planet and everything in it. And throughout all of it, life is preserved in the sedimentary record, or at least the record of life is preserved in the sedimentary record. So our rocks are like books, and they have the keys to life. And normally when you think of life in rocks, or life preserved in rocks, people think of fossils. So typically characteristic, charismatic, large dinosaur fossils. Or I like to think of these, something like this picture here, which is a really nice picture of the Torrey Sandstone, which is about 15 miles north of San Diego on Solana Beach. And there's some really fun things in this picture. You can see the bed forms on the channelized sandstone, but also there's all these little tiny, well, they're not actually tiny, circles, brown circles all around the rocks. And what these are actually fossilized burrows. These are called Ophiomorpha. And similar burrows are made by modern shrimp called callianacids in the modern tropics. But whatever shrimp like arthropod made these burrows, it has to like physically move around the sediments, right? So anything that can physically move around the sediments, or anything like a dinosaur that actually leaves like sedimentary particles like bones lying around, is a metazoan or a large animal, and those aren't really around for very long in the history of life on the planet. So they're not exactly super relevant to like astrobiology analogs using Earth, right? So what we actually care about is microbes, which take up the bulk of the biomass on the planet and also drive Earth's biogeochemical cycles. And also, luckily for us, microbes and their remains can be preserved in the rock record as well. Just in a slightly different way. These guys are typically preserved in organic matter, which gets encapsulated inside sediments and then stuck into the sedimentary record over time. So what exactly is, there we go. What exactly is organic matter and how is it preserved in sedimentary rocks? Well, organic matter is basically just everything that comprises an organism for this purpose. So we'll talk about these little green things in the middle. We'll call them algae. And these little algae are composed of different things like DNA, carbohydrates, amino acids, lipids. And over time, poor little algae will die. They'll break apart. Some portion of the algae's biomass is going to make it all the way down to the sediment or the ocean floor or whatever depositional environment they're in. And eventually it's going to get covered in sediment. And not very much of this is actually going to get preserved in the sedimentary record, about 1% at best in your average, and in some cases higher, depending on preservation potential. But overall, not that much. And while we would love to have DNA or proteins preserved in the geologic record, over the course of time, really the only things that are recalcitrant or well preserved are things like lipids, like the hydrocarbon skeletons that are on either side of the screen here, as well as characteristic isotopic signatures from life elements such as carbon, nitrogen, sulfur, etc. And these are the tools that I and my labs use to study the trajectory of life on the planet through time and try to understand what's been going on. So I'll just motivate what we do or show some motivations. So the main thing we're trying to do is make observations about ancient life from the geologic record, and actually see what was going on, what was living, what was happening, how things were making a living. This is kind of like putting together a jigsaw puzzle in time when you only have 1% of the pieces. It's kind of complex. So to kind of inform this, a lot of the times we go to the modern day And we try to look at what signals modern microbes actually leave in their environments, if they're distinguishable, if they're unique, and if they're long lasting. If we can distinguish those and then ultimately hope to track them into the sedimentary record, and then by proxy into the geologic record, how can we better interpret and understand geobiological signals preserved across geological time? So that's the ultimate goal. And then Nathan said it's a really short presentation, so I'm just gonna throw some data at you really quick of things I've done in the past and things we're currently working on in that, and that'll be that. So I've worked on a number of things spanning mass extinctions, climatic events, but the one I'm going to show today is just changes in an environmental parameter. This is kind of a lot that doesn't really make a lot of sense, but all you really need to know is on the left-hand side here, this is just a core of rock, so it's about 10 meters of rock. This is in a lake deposit. This is actually from New Jersey, amusingly. A little bit north of here in the Newark Basin, which is a failed rift basin. And what we were able to do in this study is we were able to use elemental parameters such as carbon, nitrogen, their stable isotopes, as well as a suite of relatively detailed lipid biomarker analyses, and map that onto the sedimentary features that we found in the sedimentary rocks. And basically figure out ways to sort of decouple and sort of understand how biology changes with physical changes in the lake, such as salinity, depth, and sort of alkalinity. So I'm not going to go into all of these little details because there's not time, but some really interesting things that we were able to use a nitrogen isotope to actually show this is not only a hypersaline lake, but it was also an alkaline hypersaline lake, which is kind of funny in the geologic record and relatively neat. And lastly, we were able to show with this kind of gamma-serine peak here, which is a particular C30 lipid biomarker. It's this number 4 one on the chromatograms over here. It's a biomarker of stratification. And as you go up through this lake level, as the lake is actually filling and then starts to evaporate, the biomarker gamma-serine just kind of peaks to massive dominance in the chromatograms. It's more than I've ever seen before, which is like a signal of really strong evaporation. So this is a really, really good example of when you do something in the geologic record and it turns out way better than you expect, like with the signals. This is a really fun study. But that's like all the past work. In the current, I'm part of the Dawson lab. I work with Kat Dawson. And what we're trying to figure out right now is we're looking at modern sea to shore transitions. In particular, we're looking at iron, sulfur, and carbon links, ostensibly with the goal to understand what warming worlds and rising sea levels are going to do to the terrestrial methane cycle as you bring in salty, sulfate-rich waters and flood freshwater systems. But the overall goal is to actually try to figure out what sort of lasting molecular and isotopic effects these transitional settings or transient settings have on the transient sediments that are passing through them on their way to their final depositional environment. And what we basically do as a little workflow is we go out into some rivers around the area that have like estuaries. We go to various places like the Raritan River and more pristine rivers, like these are down in the Pinelands. This is the Molokai River and its associated salt flats. We take some nice sediment cores, we process the pore water, the environmental variables, we split the core, cut the core, do DNA measurements, start incubations with whatever we feel like doing. And this is extremely a work of progress. But we look at coupled iron sulfur cycles. We look at the variation of microbial community as a function of salinity and depth. And then we also track the thanatrophy with isotopes and look at the metagenesis, trying to understand like what these systems are doing.
Who's calling?
That's basically